Okay, so continuing back with our complex carbohydrates, we talked before about how many of the polysaccharides, especially starch and glycogen, are used to store energy kind of for the medium term. When we talk about energy storage, there's really about three different scales we're looking at. We're looking at really, really short-term energy storage. You're going to find that with ATP, which is a nucleic acid, right? If you're looking at medium-term energy storage, which could be minutes, hours, maybe, you're looking at things like carbohydrates, like starch or glycogen. Glycogen if you're an animal, starch if you're a plant. If you're looking at really long-term energy storage, which is days, weeks, months, years, then you're looking at lipids or fats, those kinds of things, right? So medium term, we're looking at polysaccharides, which again are created using dehydration synthesis, right? So plants, again, use starch. Starches in plants typically are stored either in seeds, which gives the embryo a source of energy to use as it's beginning to grow, and also in the roots where it's safe to store starch below ground. In plants, you typically find a lot of starch in the roots as well. If you think about things like potatoes and sweet potatoes and carrots, those are all root crops that contain fair amounts of starch. Animals, on the other hand, use glycogen. It's very similar to starch, a little bit different in some very important ways, but it's a way that we use to store energy in the medium term. And it's made the same way. It's glucose molecules strung together using dehydration synthesis. If you're looking at polysaccharides used in structural support, then you're looking at cellulose, which is what you find in the cell walls of plants, and also chitin, which is what you find in the cell walls of fungi, and also in the exoskeletons of arthropods, which include things like insects, spiders, ticks, mites, millipedes, centipedes. Those are a lot of the arthropods that have chitin in their cell walls. Now, the interesting thing about these is they also contain glucose, but the glucose is a different type of glucose than we find in glycogen or starch. So even though it's a similar molecule, it's indigestible to most animals. When you consume cellulose or when you consume chitin, you actually do not digest it. And there's very, very few animals on Earth that can even come close to digesting it. And when they do, they have to have microbial help to do that. Right, so we talked about starches being stored in plants, upwards of 10,000 glucose monomers hooked together by doing dehydration synthesis, a lot of times in the seeds and roots, and they're broken down into glucose. So if you take a starch and do the opposite reaction from dehydration synthesis, the hydrolysis reaction, you break down the polysaccharide into monosaccharides. Glycogen, very similar. You take a few molecules of glycogen, run them through several thousand hydrolysis reactions, and you get a bunch of glucose molecules back again. Right? So again, when we have extra glucose in our bodies, by the way, this is where insulin comes in, as insulin helps our bodies to remove glucose from our blood and store it up as glycogen in our skeletal muscles and also in our livers to help get it out of the bloodstream. Glycogen can be broken down again by a different hormone called glucagon, which re-releases glucose back into the blood where it can be transported throughout the body. Right? So these are polysaccharides. Cellulose, which again is a structural, indigestible to animal kind of polysaccharide, versus starch and glycogen, which are more animal forms of. Sorry, glycogen's animal, starch is for plants, forms of energy storage of glucose. All right, so the next category we're going to look at are lipids. Lipids are much more diverse in form and function than carbohydrates are. And the one thing that really makes them work as a group or lets them be classified reasonably, logically as a group is the fact they're water insoluble. They're hydrophobic. And the reason for that is they are nonpolar molecules. Right? These molecules contain lots of carbons, lots of hydrogens, and very little oxygen, which means they're going to be nonpolar, therefore water insoluble, aka hydrophobic. Right? So these are going to be not things that dissolve very well in water. Carbons and hydrogens share electrons equally, so those covalent bonds are nonpolar, which means they don't like interacting with water very much. So we're dealing here with things like fats, oils, waxes, and some vitamins, which we'll talk about a little bit here in a few minutes. Another really important kind of lipid for our purposes is the phospholipid. And phospholipids are unique in that they actually do contain part of a molecule which is hydrophilic and also part of a molecule which is hydrophobic. So they're actually both hydrophilic and hydrophobic at the same time. The reason when we talk about phospholipids they're the most common molecule that we find in the plasma membrane of cells. So the barrier that separates the inside of the cell from the outside of the cell, which is the plasma membrane, is made in large part by phospholipids. Right? So steroids are another type of lipid that we see, and these are simply just carbon rings fused together to give them, again, a very much water-insoluble characteristic, but a lot of times are things like testosterone and estrogen, which are male and female sex hormones, respectively. Right? So this picture down here at the bottom of the slide really nicely illustrates, I think, the fact that this is going to be a nonpolar molecule. So if you take a look at all of these carbons and all of these hydrogens, and when you think about the fact that all of these straight lines represent nonpolar covalent bonds, this molecule is going to be very much nonpolar, hydrophobic, water insoluble. despite the fact that it has a carboxyl group down here at the end, which would tend to make this end of it slightly hydrophilic, the fact that the rest of it is so big and so water insoluble means this molecule would actually be hydrophobic. Some of the things lipids do, they're involved in compartmentalizing the cell, like we talked about with the plasma membrane, but even other membranes inside the cell, like you're going to see with the nuclear envelope and the ER and the Golgi and vesicles and lysosomes, mitochondria, chloroplasts. All of those things have membranes around them, which are made out of phospholipids. Right? We see lipids being used in energy storage, like I talked about previously. Fats and oils are really good ways to store energy for the long term. Days, weeks, months, years kinds of time scales are going to use lipids to store up energy because they're very energy dense. You get a lot of calories in a relatively small volume, so we say they're energy dense, very efficient ways to store calories. They're also very good at insulating, so thermoregulation or maintaining the body temperature. Using lipids, fairly good way to do that. They also provide protection, right? So lipids are involved in cushioning support around some of our internal organs. That if you take a blow to the kidney, for example, there's a layer of fat around those kidneys and some connective tissue as well, other kinds of connective tissue as well, to help protect them. It's also a protective function. And also a lot of the hormones are lipid-based too, so it's involved in communication within the body as well. Some lipids, at least, are also very good at waterproofing. We see various oils used in certain types of aquatic mammals. We see waxes being used in plants and insects as well. So they're a very diverse kind of polymer. So the main one that we're going to focus on for this lecture is going to be the glyceride. Right? And so glycerides, primarily triglycerides, are the main component, dietary component of what we get with the fats we experience every day. So a triglyceride is going to consist of a single glycerol molecule, which is an alcohol, and therefore is hydrophilic, and 3 fatty acid tails. So by taking 3 fatty acid tails and 1 glycerol and doing some dehydration synthesis reactions, we create a larger triglyceride molecule. So down here, you can actually see the structure of this. This was the glycerol. These are the fatty acids. They're hooked onto the glycerol to make a larger triglyceride. Now, the thing about these fatty acid tails is that they don't all have to be the same. Some of them can be shorter than others can be longer. Some of them can be saturated, others can be unsaturated. So a saturated fat refers to the fact that all of the carbons are completely surrounded by hydrogens and only consist of carbon to hydrogen bonds. There are no carbon to carbon double bonds of any kind anywhere in the molecule. That makes something a saturated fat. Saturated fats typically, most of the time, come from animals, and they're solid at room temperature. Right? So lard, for example, is a type of saturated fat. At room temperature, it's solid, and lard, of course, comes from animals. Okay? As far as unsaturated fats, they tend to contain 1 or more carbon-to-carbon double bonds, which is going to help make them liquid at room temperature, and these are going to primarily come from plants. Right? So the one exception that we're thinking of here would be coconut oil, which is an oil, which is plant origin, but actually happens to be a saturated fat. That one's kind of an oddball, but the rest of them, oils are liquid, fats are solid, oils are from plants, fats are going to be from animals. Sorry about that. So there are going to be some important effects on health here as well. Unsaturated fats tend to be a little bit healthier than saturated fats. And the more of those carbon-to-carbon double bonds you have, the healthier it tends to be as well. You've also probably heard of trans fats. And trans fats are an artificial type of fat that's created for various purposes, whether it's shelf life or taste or these kinds of things. They're created industrially to create some desirable effects on the food from a marketing standpoint, but also have some very negative impacts when it comes to our health. Right, so for example, up here we have a glycerol molecule up here, and we have 3 different fatty acids. So if we pull out 1, 2, 3 water molecules, 3 are taken out, that's dehydration synthesis, we make a triglyceride. Now this triglyceride is made up of exclusively carbon-to-carbon single bonds, which makes it a saturated fat. This would be solid at room temperature. This would be like a fat that we find in butter, for example. On the other hand, over here, we have another triglyceride where this tail has 2 carbon-to-carbon double bonds, which makes it an unsaturated fat. And because of these carbon-to-carbon double bonds, that's going to make this one a liquid at room temperature and help to remind us that this is going to be, in fact, from a plant and not from an animal. On the other hand, if we want to go back, if we want to take our fats and break them down, we take 3 water molecules, put them into the molecule this way, we break down our triglyceride back to our glycerol and our 1, 2, 3 fatty acid tails. So again, dehydration synthesis, hydrolysis, it works for all these molecules. It works for carbs, it works for proteins, it works for lipids, it works for nucleic acids. Same reaction for all these things, as well as for hydrolysis as well. Right? So this is, again, is a saturated fat. All these carbon-to-carbon single bonds, all these H's surrounding those carbons makes this entire thing a saturated triglyceride fat. So not especially healthy, but animal in origin. Right? So we see these a lot in fatty meats. We see them in dairy products. We see them in coconut oil, like I mentioned a few minutes ago. So by taking this fatty acid tail, the two other ones, hooking into a glycerol, you would get a saturated fat. On the other hand, a monounsaturated fat would involve a single carbon-to-carbon double bond. So you'd have this fatty acid plus two other ones plus a glycerol to give you a monounsaturated fat. So olive oil, canola oil, and peanut oil are all good examples of monounsaturated fats. And the effect of this carbon-to-carbon double bond is that these fats are healthier than saturated fats. Kind of a simplistic way of looking at it or thinking about it is that when you have a whole bunch of carbon-to-carbon single bond tails like these fats pack in nice and tight and close together. They can form blockages, they can form all kinds of nasty things in the body, which can create some difficulties later on in life. The carbon-to-carbon double bonds make the tails kinkier, bendier, less likely to pack in close together and form those blockages like we saw with a saturated fat. So they're going to be a little healthier. This actually would be a polyunsaturated fat because we have on this picture 1, 2, 3, 4, 5, 6 different carbon-to-carbon double bonds. All kinds of bends and kinks and twists in these tails means that these fats are going to have a much harder time packing in close together, less likely to form blockages, so a healthier type of fat overall to consume in our diets. Polyunsaturated fats are things like corn oil and sunflower oil. Those are two fairly common polyunsaturated fats. So again, if you take this fatty acid, take two other ones, hook it to a glycerol, you would create a polyunsaturated fat. So saturated solid room temperature, unsaturated is liquid at room temperature. Saturated from animals, unsaturated from plants. All right, so getting into the idea of trans fats for a moment, just because we've probably all heard of those, trans fats are produced industrially through a process called hydrogenation. So anytime you see a product that contains partially hydrogenated anything, you know you're dealing with a trans fat. All right, so the way that they do this is they take plant oils and they expose them to high levels of hydrogen gas. Some of those hydrogens stick to the carbons. The carbon-to-carbon double bonds are broken, and carbon-to-hydrogen single bonds are formed, and that creates a saturated fat from a previously unsaturated fat. So, kind of a cynical way of looking at this is that we're taking healthy things and turning them into unhealthy things by doing hydrogenation and creating trans fats. The reason why this is done is that trans fats tend to be creamier in texture than oils. They also tend to last longer on the shelf. So from an economic standpoint, they're better, I guess you could say, than using oils, right, kinds of things. So by taking partially hydrogenated corn oil, turning it into a saturated fat, you've created something which is cheaper than animal fat, lasts longer on the shelf, tastes probably better as well, but has also some relatively negative effects on the body. In particular, trans fats have been shown to increase the LDL levels in the body, and LDL is your low-density lipoprotein, so-called bad cholesterol. It tends to increase levels of bad cholesterol and decrease levels of good cholesterol, which is the exact opposite effect that you really want to have on those molecules. All right, so trans fats, like I said, increase LDL and decrease HDL. They increase LDL, decrease HDL. They increase the bad, decrease the good. Cholesterol, by the way, is something which we do need to have in our diets, at least in low levels, because it's a precursor to things like testosterone and estrogen and other steroid hormones. So we do need to have some, but consuming things like this picture up here, which, trying to think of something which could contain trans fats, and a lot of them, this is what came to mind. It's a KFC Double Down sandwich, which has been around, I think it's around anymore, but was around for a few years, and very likely contained huge amounts of those fats. Cholesterol is important to have in small quantities, but certainly in larger quantities might not be such a good thing to consume a lot of. I mentioned before how fats and oils can also be used to store energy for the long term. Because they're relatively stable, because they're relatively energy dense, you find a lot of organisms that use fat to store up energy. All right, so this bear, which has been consuming all kinds of food, probably all summer and early fall, in an attempt to fatten up for winter, has huge numbers of calories stored up in its body in the form of fat. All right, so if you break down fat, if you take roughly a pound of fat, from an energetic standpoint, that's worth about 6,000 calories. And so if you wanted to store 6,000 calories worth of carbohydrate, you would need to have a much larger volume of carbohydrate to do that, which means effectively this bear would be 30% bigger, which it's already kind of big. It's sort of scary. I wouldn't want to see this bear 30% bigger. But regardless, fats are a very good way to store up energy. All right, waxes are the most hydrophobic of all the lipids. They contain extremely long chains of hydrocarbons. Carbon to hydrogen single bonds, which of course are nonpolar and therefore hydrophobic, are always used for waterproofing. Right, so these are very saturated, very waterproof. We tend to find them on the stems and leaves of plants. where it waterproofs them, not so much to keep water out, but to keep water in. Right? So living on land for any organism, whether a plant, an animal, or otherwise, creates a challenge of drying out, desiccation. And so by sealing the plant off with wax, you help to keep water on the inside. Now, there's also a downside to doing that, because if you seal off the plant against water loss, you're also potentially sealing it off against gas exchange, but there are ways around that, which we'll see later on in a future chapter. A lot of aquatic mammals and birds have oil glands or wax glands they use to waterproof themselves as well. In this case, it's used to keep the cold water away from their skin, where it might serve to pull heat away from the body. In this case, though, it's going to help waterproof them. And insects, too, have a waxy layer called a cuticle on their exoskeleton, which, too, helps them seal in water and prevent them from drying out. And, of course, bees use them as well in honeycombs. I mentioned phospholipids a little while ago. Phospholipids consist of a hydrophilic head and a hydrophobic tail. Right, so in these molecules, the heads are the polar water-soluble parts, the tails are the nonpolar hydrophobic water-insoluble parts, and so the reason why these work this way is the phospholipids have a phosphate group associated with the hydrophilic head. The phosphate group is very strongly polar and therefore confers a hydrophilic nature to that part of the phospholipid molecule. Right? So when we're looking at phospholipids individually, one of these molecules has a hydrophilic head and two hydrophobic tails. Okay? And so when you put these in water, which is a hydrophobic environment, they're going to arrange themselves spontaneously into a bilayer, which effectively is what a plasma membrane or any other cell membrane is about, is being a phospholipid bilayer. So we see those very commonly, and they're very important in terms of compartmentalizing cells into parts of cells that do different jobs. Okay, so if we have an experimental setup like this, where we have water, which is water soluble, and oil, which is water insoluble, phospholipids, which are both soluble and insoluble in water, line themselves up this way. The hydrophobic tails go to the oil, the hydrophilic heads go to the water, and they form, in this case, a single layer because the oil is floating on top of the water. They're able to do that. Bilayers form when phospholipids are placed into pure water, and the reason this occurs is so the hydrophobic tails don't have to come into contact with one another. Right? So all these hydrophobic tails are mildly attracted to each other, but very strongly repelled by water, and the hydrophilic heads are attracted to water. So they're going to point out to the water, whereas the tails point inwards toward each other. Right? So that creates a bilayer. Steroids, like I mentioned a few minutes ago, also are carbon-to-carbon rings that are fused together, typically 4 of those rings. A lot of these start off as cholesterol, but then later on get turned into things like estrogen and progesterone, which, outwardly look very similar, but of course functionally do very, very different kinds of things depending on, your personal persuasion as far as whether you produce estrogen as a male or testosterone as a male or estrogen as a female, which are the two sex hormones. Testosterone for males and estrogen for females. Pharmaceutical steroids, when you hear about steroids in kind of a negative context, are a different story. They're used to do other kinds of illicit activities, but natural steroids are perfectly normal, perfectly common to have in the body, and very important from that standpoint as well. All right, so we're going to cut off here before we start up with proteins, and we'll do proteins as a separate section as well.